Dear students, the topic is biosensor.
Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Biosensors are analytical devices that consist of biological detecting elements like a sensor system and a transducer. When we compare with any other presently existing diagnostic device, these sensors are advanced in the conditions of selectivity as well as sensitivity. The applications of biosensors mainly include checking ecological pollution control in the agricultural field as well as food industries. So a biosensor consists of a biological element, maybe an enzyme, a nucleic acid, otherwise an antibody. The bio element communicates through an analyte being checked, and the biological reply can be changed into electrical signal using transducer. Based on the application, biosensors are classified into different types like resonant mirrors, immune, chemical canaries, optrodes, biocomputers, glucometers and biochips. The first biosensor was invented in the year 1950 by an American biochemist L.L. Clark. This biosensor is used to gauge oxygen in the blood, and the electrode used in this sensor is named the Clark electrode or oxygen electrode. A biosensor includes two main distinct components like biological components such as cell, enzyme and a physical component like an amplifier and transducer. The biological component identify as well as communicate through the analyte for generating a signal that can be sensed through the transducer. The biological material is properly immobilized over the transducer, and these can be frequently used numerous times for a long period.
The main components of a biosensor are three. One is sensor, is a responsive biological part. Two is transducer, is the detector part that changes the resulting signal from the contact of analyte, and for the results it displays in an accessible way. Third one is associated electrons, comprises an amplifier which is known as signal conditioning circuit, a display unit as well as the processor. Working of biosensors. The electrical signal of the transducer is frequently low and overlays upon a fairly high baseline. Generally, signal processing include detecting a position baseline signal obtained from a related transducer without any biocatalyst covering. The comparatively slow character of the biosensor reaction significantly eases the electrical noise filtration issue. In this stage, the direct output will be an analog signal. However, it is altered into digital form and accepted to a microprocessor phase where the information is progressed. Influence to preferred unit and output to a data store.
Next, we can see the evolution of a biosensor. The classification of biosensor can be done into three generations based on the amount of incorporation of the separate component like the technique of connection of the bioreceptor molecule, otherwise biorecognition towards the element of the base transducer.
In the first generation, the molecule of the bioreceptor is entrapped physically within the area of the base sensor after a discriminating membrane like a dialysis membrane. In the next generations, the achievement of immobilization can be done through covalent bonds on a properly customized transducer interface, otherwise by inclusion into a polymer matrix on the surface of transduction. In the second generation, the individual components stay separate like control electronics, biomolecule and electrode. In the third generation, the molecule like bioreceptor turns into an essential element of the base sensing element, whereas these definitions were possibly planned for enzyme electrode systems. Related classifications are suitable to biosensors usually can be made. It is within the second and third generations of families main development attempt can currently be observed.
There are different types of biosensors and are classified based on the sensor device as well as the biological material.
Generally, the electrochemical biosensor is based on the reaction of enzymatic catalysis that consumes or generates electrons. Such type of enzymes are named redox enzymes. The substrate of this biosensor generally include three electrodes such as a counter, reference and working type. The object analyte is engaged in the response that happens on the surface of an active electrode, and this reaction may source also electron transfer across the dual layer potential. The current can be calculated at a set potential. See, the diagram is given. Electrochemical biosensors are classified into four types: amperometric biosensors, potentiometric biosensors, impedimetric biosensors and voltammetric biosensors. Amperometric biosensor is a self-contained incorporated device based on the amount of the current ensuing from the oxidation, offering exact quantitative analytical information. The simple amperometric biosensor in frequent usage include Clark oxygen electrode. The principle of this biosensor is based on the amount of the flow of current between the counter electrode and the working, which is encouraged by a redox response at the operational electrode. Choosing analyte centers is essential for a wide selection of uses, comprising high throughput medicine screening, quality control, problem finding as well as handling and biological checking. Potentiometric biosensor provide a logarithmic reply by means of a high energetic range. These biosensors are frequently complete by monitor producing the electrode prototype lying on a synthetic substrate covered by a performing polymer with some enzyme is connected. These comprise two electrodes that are enormously responsive and strong. They allow the recognition of analyte on stages before only attainable by HPLC, LCMS and without exact model preparation. Impedimetric biosensor is a responsive indicator for a broad range of physical as well as chemical properties. The technique of impedimetric have been executed to differentiate the invention of the biosensors as well as to examine the catalyzed response of enzyme, lectins, nucleic acid, receptors, all cells and antibodies. See, diagrammatic representation is given here. Voltammetric biosensor. This communication is the base of a new voltammetric biosensor to notice acrylamide. This biosensor was built with a carbon glue electrode customized with a hemoglobin, which include 4 prostatic groups of the heme, that is iron. This type of electrode shows a reversible oxidation or reduction procedure of hemoglobin. See, the diagram is shown here.
Next is a physical biosensor. In conditions of classification, the physical biosensors are the most fundamental as well as broadly used sensors. The main idea behind this categorization also happened from inspecting the human minds. As the general working method behind the intelligence of hearing, sight, touch is to react on the exterior physical stimuli. Therefore, any detecting device that offers a reaction to the physical positions of the medium was named as physical biosensor.
The physical biosensors are classified into two types: piezoelectric biosensors and thermometric biosensors. Piezoelectric biosensors are a collection of analytical devices which work on a law of affinity interaction recording. The platform of a piezoelectric is a sensor element that works on the law of oscillations transformed due to a collection gem on the surface of a piezoelectric crystal. In this analysis, biosensors having their modified surface with an antigen or antibody, a molecularly stamped polymer and heritable information. The declared detection parts are normally united by using nanoparticles. Thermometric biosensor. There are various types of biological reaction which are connected with the invention of heat, and this make the base of thermometric biosensors. These sensors are usually named thermal biosensors. Thermometric biosensor is used to measure or estimate serum cholesterol. As cholesterol obtains oxidized through the enzyme cholesterol oxidase, then the heat will be produced which can be calculated. Similarly, assessment of glucose, urea, uric acid and penicillin G can be done with these biosensors. Optical biosensors are devices that uses an optical measurement principle. is a device. Optical biosensor is a device that uses an optical measurement principle. They use fiber optics as well as optoelectronic transducers. The term optrode represents compression of the two terms optical and electrode. These sensors mainly involve antibodies and enzymes like the transducing elements. Optical biosensors permit a secure, non-electrical, inaccessible sensing of equipment. An extra benefit is that these frequently do not need reference sensors because the comparative signal can be produced by using a similar light source to the sampling sensor. The optical biosensors are classified into two types, namely direct optical detection biosensors and labeled optical detection biosensors. Wearable biosensors. The wearable biosensor is a digital device used to wear on the human body in different wearable systems like smartwatches, smart shirt, tattoos, which allow the level of blood glucose, BP, the rate of heartbeat, etc. See here, wearable biosensors are given. Immunosensors. Immunosensors were recognized on the truth that antibodies include high affinity to their particular antigens, like the antibodies particularly combine to toxins or pathogens or interact through host immune systems component. These type of biosensors are based on affinity ligand solid state devices where the reaction of immunochemical can be connected to a transducer. Magnetic biosensors are used to gauge changes within magnetically persuaded effects of magnetic properties. These kind of sensors use crystals or particles of super paramagnetic, otherwise paramagnetic, to detect biological communications through measuring changes within magnetic properties like changes within coil inductance resistance. See, magnetic biosensors design is given here. Resonant biosensors. In a resonant biosensor, a transducer like an acoustic wave can be connected through a bioelement. Once the analyte molecule is connected towards the membrane, then the mass of the membrane alters. So the final change within the mass subsequently alters the transducer's resonant frequency. After that, the change in frequency can be measured. Thermal detection biosensor. Thermal detection type biosensor uses one of the basic biological reaction properties like heat production or absorption and changes the temperature when the reaction occurs. The designing of this sensor can be done by utilizing the molecules of an immobilized enzyme using temperature sensors. Once the analyte and the approaches in contact, then the enzyme's heat reaction can be measured and adjusted against the concentration of the analyte. The whole heat generated, otherwise absorbed, can be proportional towards the molar enthalpy and the total number of molecules within the reaction. The temperature measurement is normally achieved through a thermistor known as enzyme thermistors. Thermistors are ideal in some applications as they are sensitive to thermal changes. Not like other type of transducers, thermal sensors do not require regular recalibration and they are insensible to the properties of electrochemical and optical of the sample. These sensors are used to detect pathogenic and pesticide bacteria.
Next we can see the applications of biosensor. Biosensor devices include a biological element as well as a physicochemical detector, and the main function of this device is to detect analyte. So the applications of biosensors are in a wide range. These devices are applicable in the medical, food industry, marine sector, and as they offer good sensitivity and stability as compared with the usual techniques. In recent years, these sensors have become very popular and they are applicable in different fields.
Biosensors are applicable in various fields like prosthetic devices, drug discovery, disease detection, environmental monitoring, soil quality monitoring, food quality monitoring, toxins of defense interest, water quality management, common healthcare checking, metabolite measurement, screening of sickness, insulin treatment, clinical psychotherapy and diagnosis of disease, in military, agriculture and veterinary application, drug improvement, offense detection, processing and monitoring in industrial ecological pollution control, diagnostic and clinical, industrial and environmental applications, study and interaction of biomolecule, development of drug, detection of crime, medical diagnosis, monitoring of environmental field, quality control, process control in industries, pharmaceutical manufacturer and organs replacement. Thus, biosensors are becoming gradually more complicated, mostly due to a blend of advances in two technical fields like biotechnology and microelectronics. These are highly important devices to measure an extensive spectrum of analytes like gases, organic compounds, bacteria, and ions.
Dear students, thank you for watching. See you in next class with another topic. Don't forget to leave a like and please subscribe my channel.